Hello everyone, today is the first week. We will learn about the specimen collection lab. The learning outcome is to understand how to collect the specimen for laboratory diagnosis. Second, know how to process with the specimen and identify using microscopic and macroscopic examination. Third, explain how to decide to accept or reject that specimen and report the results and interpretation. And the last one is know how to identify yeast in clinical specimen. So our specimen today is sputum. So here is the materials and reagents that we're gonna use for today examination. So we have this sterile screw top container, and we have the gram stains reagent, glass light, loop needles, microscope, immersion oil, and this is a agar that we're gonna use, which are blood agar, chocolate agar, and macaroni agar, candle jar, and incubator. For the procedures, we have first a sputum collection, direct examination for gram staining, bacterial cultivation, and gram staining from bacterial colony. Okay, for the sputum collection we're gonna do today is then first you need to collect your container and label it with the name, surname, type of specimen, date of storage on the side of the container. After that, you will wash your mouth with the clean water which we provided you, and then open the container lid. So after that, you can inhale and exhale deeply for 2 to 3 times, and after that, then you breathe in strongly, so your body will automatically cough deeply. And at that time, you place open container close to your mouth to collect the specimen, and please try to make that specimen, your sputum, go inside the specimen collection directly and close the lid tightly. So at the end, you wash your hands and collecting this after the collecting specimen. So after that, that sputum will be ready for the examination. After getting the sample, the next thing that we're gonna do is direct examination, which is gram staining. So we use the wood sticks or skewer, and we break it into 2 pieces. Then we use the broken ends to pick up the sputum and smear it into the center of the slide 2 by 3 centimeters thinly. So after that, we wait for smear to be dry and heat, fix, and then do the gram staining. Then we do the report and interpretation to set if this specimen is accepted or rejected. So what we're going to report is number of the bacterial cells, number of PMN stands for polymorphonuclear cells, including neutrophil, basophil, and eosinophils. And for the number of the squamous epithelial cells, these PMN and squamous epithelial cells we observe in low power field, but for the bacterial cells we use the high power field. Okay, for reporting the number of cells from gram stain, we observe for PMN and SEC 20 low power field, and we can average it. Then we count that cells per low power field. So we can report as, like, if you observe less than 1 cell, you can report it as rare. And 1 to 9 cells report as few. 10 to 25 cells report as moderate, and more than 20 cells you can report as numerous. It's the same goes as PMN and SEC. And for bacterial cells, we observe in oil field, which is 20 to 40 oil field. And if you observe one cell, you can report as rare. If you observe 1 to 5 cells, you can report as few. Report 6 to 30 cells, report as moderate. Observe more than 30 cells, you can report as numerous. So if your specimen contains a lot of contaminated saliva and found PMN cells less than 25 cells per low power field and SEC more than 10 cells per low power field, means that this specimen is not proper and it's not the sputum, so this can be rejected. This is the report example. You can report it as numerous gram-negative bacilli, few gram-positive cocci in short chain, few SEC, and numerous PMNs. Or you can report it as bacteria not seen or bacteria not found. For bacterial cultivation, you will be given CA, BA, and McConkey agar. CA is chocolate agar, BA is blood agar, and MC is McConkey agar. You can smear it respectively from CA, BA, and MC in the 4-plane technique. And after that, CA and BA, you can culture it in candle jar at 37 degrees Celsius for 24 hours. For the McConkey agar, in the ambient air, 37 degrees Celsius for 24 hours as well. So when you report it, you can report it as here. For example, growth on just only first agar plane, you can report it as rare. Growth on second agar plane, you can report it as few or light growth, or you can use the few. It's I think it's quite easier. For the third agar plane, you can report it as moderate, and fourth plane can be numerous or mini or heavy growth. And thank you for your attention.